Okay, we've submitted our binning jobs. Now we want to look at some of the results. There are a number of ways you can get to a completed job in BV-BRC. You could go to the jobs page by clicking on the jobs monitor down here at the right. You can click on the workspaces tab, which opens up the drop-down box. Now you could navigate to it by going to your home genome, or you might have shared it with someone, so that would be in workspaces, and look from there. Or you could go up here to my jobs. So we click on that and that opens the jobs page, and I've run a lot of jobs. So you can see all the jobs that I've run over time. And I don't want to sort through all of them. So I'm going to click up here on all services, and then I'm going to go all the way down to Metagenomic Binning and click on that. And that is going to show me the 631 jobs that I have submitted to this service. And you can see that I've had my successes and I've also had my failures. I'd like it to all be 100%, but it's not. So let's look at one of these samples from the one that we submitted, which is from that street bioinformatic job that we looked at from the people who had those joint replacements. Here's one of those here. It ended in 262. So I'm going to click on view that, and that opens the results for this particular job. Let's increase it a bit so we can see it more easily. This is what the jobs page looks like for the metagenomic binning job. Note that you have two binning reports here. One is for the bacterial binning report and one is for the viral binning report, because when I submitted this job, I said I wanted both. Also notice the information about the job, like the job number, how long it took to complete. And if you click on the parameters box, it would show you exactly what you submitted. In this particular video, we're just going to go over the bacterial binning report, and then we'll do a separate one for the viral binning report, and then we'll go into all of this other stuff. So I'm going to break it up because there's a lot of information here. So bacterial binning report. So we click on that. And you're old hand at this now. You know, when I click on a row, it's going to open up the green action bar and show you things that you can do downstream. So I can view it here or up here in the top right, but I like that green background. It makes me think of a nice green eye, so I'm going to click on that. This is the binning report for our sample. And it gives me some of the information about it. It says it found one good bin and 0 bad bins, and we'll describe in more detail what that means. And then it gives you this table. Let me give us some more real estate here. Note that it says this. These are the things that establish if it's a good bin or a bad bin. So it's got to have a completeness of over 80%, a fine consistency of greater than or equal to 87%, a contamination of less than 10%, and have a single phenylalanine tRNA synthase gene, which is just the gene that the program, the people chose who designed this algorithm as something that almost all bacterial genomes have, all should have it, and they should only have one copy. So if you have that, it has a yes. But let's go into the details of what each of these column heads mean. The score. This is kind of an odd one. It's a weighted score of the completeness and consistency scores. So the completeness and the consistency, subtracting the contamination. Okay, so that's just what the developers decided was a good thing, but let's talk a little bit about what a score means. If you have something that's heavily contaminated, this could end up to be a negative score, which tells you it's not any good. But the range of this number goes between 2090 and negative 5,000. Okay, so I don't pay much attention to the score. I pay attention to more of these other things. The genome ID is the unique identifier assigned to our binned genome. And here's the name of our binned genome. Notice it says Haemophilus influenzae clonal population. It's just telling you when it gives it a genus and a species, it's saying out of all the reference genomes that are used in this pipeline, this was the closest one to the bin. So when it was comparing our data to all the genomes that are backed in this service, the closest one it got was Haemophilus influenzae. If you want to see the reference genome, this is a hyperlink that will lead you there. So let's talk briefly. I would encourage you to look at the tutorial. Some of these things are pretty wordy for each of these things. The coarse consistency, if it's high, like this one is, 99.3%, that's an indication that the bin is either a single genome or it could be that there were several closely related genomes in there that they're so close to each other that you can't separate them. Like maybe you're streaking something out on a plate and you've gotten it pretty well down, but you might have picked up two things that are the same thing, maybe a few snips difference. It can't distinguish between those. Fine consistency. If the number is high, as we mentioned earlier, it indicates a good bin. And so this is an indication of that. Completeness and contamination. Both of these scores are determined by two different algorithms that the programmers developed, eval-G and eval-Con. Now, I don't want to go into the details of the mathematical formulations underneath those, but if the genome is sufficiently complete, above 80%, we've got a good genome. If it's contaminated less than 10%, we have a good genome. So far, everything's good. Now, hypothetical. This is something you should think about. All bacterial genomes have a number of hypothetical proteins. They're assigned that as hypothetical. It's got a start codon, it's got a stop codon, it's got a certain sequence length, but we don't know what they do. And we don't know if they're even produced and actually do anything. So anything that looks like that, it gets hypothetical. And like I said, all bacterial genomes have it, but they have a certain number. I generally think around 30% of a lot of the genomes have hypothetical genes. If it was a lot higher than that, if it was 50%, 80%, I would start worrying about it, and I would indicate a poor quality genome. Contig count is pretty explanatory. That's how many contigs were assembled in the genome. The size of the predicted genome from this bin. Contigs N50. This is something they talk about in assembly all the time. And this is the size of the largest contig such that half the base pairs are in this contig size or larger. So it's just a measure of how good it is. If you have a lot of really, really, really small contigs, it's an indication that something is wrong. Coverage. We've talked about this before. Coverage indicates how many reads are mapped to these individual contigs. And if there's no coverage information, it'll just say 50 for some reason for every bin. Potentially problematic roles. It's looking at all the genes or proteins that are annotated and looking for things that don't look right. So across all the genes that are annotated for my binned genome, it found that 16 had a problem, and it has a good phenylalanine tRNA synthetase. Now, generally, when you have a problem, one of these cells is yellow. None of these have problems. Note that anything that's blue, score, genome ID, reference genome, potentially problematic roles, They're underscored, which means it is a hyperlink. So if I click on this 16 roles, it will take me to a page that shows me what genes are potentially problematic. Now, what it first opens is the genome report, and these are the statistics on the bin genome. So I have to scroll down past that. And here are the potentially problematic roles. So the role is the function that it thinks this gene has. The predicted count is what the pipeline thinks should be annotated in this particular genome. The annotated count is the ones that it actually found. So if I was able to find one, there would be a feature link that would take you directly to that problem gene. And there's a comment then that tells you some information about it. So I want to start with something that for me is a little easier. For example, let's do this particular gene, and I'm not even going to try to pronounce it. But the pipeline predicts that there should be one of these genes. And actually it found two. And so this would take me to a table that showed me those two genes. But this is the description. So it says this is the unique gene identifier for one of those genes. This 727.4148 is the genome ID for our bin genome. PEG stands for protein encoding gene 1590, meaning this is the 1590th gene annotated in our bin genome. And it says it's on this contig. And the contig is short. If you looked at the reference, you notice that this number is different. So it's closest to this reference genome, and they have this many matching k-mers. But this is on a short little contig. And then you have this one, which is near the end of a contig, which is an indication to me, and this gene is short. I would be looking at these things to see if it's actually an annotation problem, especially when it says it's at near the end of the contig. So it's an indication to me that maybe those contigs are meant to be together and the assembly pipeline just couldn't pull them together. Okay, so that's an easy one to understand, or, sort of easy to understand. So it's indicating an assembly error. This particular gene, the pipeline thought we should have one. There were actually two. This would link to that. And it says here that this particular gene, number 281, which is found in this contig, and it shares a lot of matching k-mers with this gene in the reference genome. But there's another one that's in a different contig. What we need to do is, if you're interested, because you can spend a lot of time digging into these things. And so generally I only spend the effort when I think it's going to lead to my understanding about the genome or give me something unique I can put into the publication. But if I wanted to see if this was a true duplication event, I would take this reference gene and these two genes and build an alignment and look at the sizes of them and see if they really should be together or if this is a true duplication event. And now we have these things. For some reason, the pipeline says we should have one. It couldn't annotate any of them, and we don't have it, so we can't point to it. So that's kind of a harder one to understand. So that is what the problematic roles gives you. So I'm clicking the back button because I want to go back to that report. This would take me to the genome that was the reference, and this takes me to the exact same place that the potentially problematic roles do. So that and the score lead to that genome report page. This leads to the reference genome. This leads to our genome landing page. And this will show me it's already been indexed in the resource. You can put it in the trees. You can use it in the variation service. It's all there for you. And there's that unique identifier. And here is all the information about that genome, including the coding sequences. 1,808 genes were called in our bin genome. 174 of them were annotated as hypothetical. So that's where all that information is, putting it up in a nice, neat bundle in the genome report. Now, what we didn't get to see was what a bad bin looks like. So let's see what that looks like. This doesn't have anything to do with antimicrobial resistance, but this is a great sample and one I'm very fond of. This is in the Sequence Read archive, and I found it. It's from a squished tsetse fly, which is a fly who's a vector of disease, specifically sleeping sickness, in Africa. And they just squashed some of these flies, and I used that as an example because I think that's so cool. So let's just look at that job. And let's look at the binning report. And we want to view that. And I'll give it some more real estate here. And it says, oh, you've got one good bin and one bad bin. And this is the good bin, and we know all about that. This had a few more problematic roles than our Haemophilus clonal population genome. And then this guy had some issues. The contamination was high, and it couldn't find BS. So that is what a bad bin looks like. But let's look at this. I'm going to click on this genome ID. So even though it's a bad genome because it was missing VS and it had some contamination, it's still in the resource. It may have had some problems, but it's still here, so you can still analyze it. Now, next, we're going to look at the viral bins. So I know you're going to want to join me for that because it's very exciting, especially the ones that are coming from this squashed African fly. See you there.